I don't want to force anybody to introduce themselves if they're not comfortable. So let's go ahead and get started. What we're going to start with this morning is a discussion of the theory of the LI 6800. And so what we do there is we want to talk about how we get the information that we see. Everybody can see my screen?
Yeah.
Excellent, thank you. Okay, so as I said, we're going to start with the LI 6800 instrument theory, which is basically how it works and what it can tell you. Because if we need to know how it all works together, it helps to understand the principles of it. So a couple of things to keep in mind. Some of the equations are going to be simplified. Okay, I do not mention a couple of terms. All of the actual terms are in the manual. So all the full equations are in the manual, which is quite large and full of information. The other thing I want to encourage is asking questions as we go. I have some stopping points in this lecture where I specify questions. The thing I would encourage you, and then Shannon will be monitoring the chat as I'm teaching. I'll be focusing on the presentation and she'll be focusing on looking at the chat. And we're going to switch it. So if she's teaching, then I will be focusing on the chat. And that way we can have both of us looking at the chat. At least one of us is always looking at the chat to make sure that if you have any questions, we can help. So let's start with the basics. For some of you, this may be a review, and you may think, oh, I know this, but for some of you, your plant physiology class might have been a couple of years ago. So let's start with talking about photosynthesis and photochemistry. The LI 6800 is a portable photosynthesis system, but we're actually measuring photochemistry, or you might hear it called photophysiology. So we have a leaf here, and you can see that we have various chloroplasts, and inside you can kind of see, we have various cells and we have chloroplasts inside of them, and we can see the stomates on the bottom of the leaf. And photochemistry is in large part transpiration, which is water coming out, and assimilation, which is CO2 going in. From that basic transpiration and assimilation, we can also start to think about the Calvin-Benson cycle, which is also called the Calvin-Benson-Basim cycle, and the photosynthetic carbon reactions. It really depends which textbook you're looking at, but CO2 assimilation is used in the Calvin-Benson cycle. And part of that CO2 is used to keep the cycle going, and then part of it goes off to sucrose and starch synthesis. The Calvin-Benson cycle requires ATP and NADPH. We need ATP and NADPH to continue that process. We get the ATP and the NADPH, the light reactions of photosynthesis. And light reactions of photosynthesis you might see referred to as the Z-scheme or something like that. What happens is light comes in to the plant, and then it goes through the reaction centers, through PS2, plastoquinone, cytochrome B6F, PS1. We have the ATP synthase always doing its spinning, and then the oxygen involving complex. You'll often see this particular Z-scheme reversed from the way that I have it on this particular slide. It doesn't really matter which direction you put it in, as long as the electrons are shown going the correct direction. In this case, it's displayed this way so that the ATP is over by the Calvin-Benson cycle. And along with light going in and along with the electrons going to photochemistry, they could also go off to non-photochemical quenching. And non-photochemical quenching is any use of the electrons that is not either photochemistry or fluorescence. And fluorescence is the other process. A lot of other processes are happening. And we are looking at the first. As I mentioned, any photon that goes into the leaf has to go somewhere. So a photon goes in, a chlorophyll molecule is raised to an excited state. That excited state is extremely unstable. And so immediately that electron wants to fall back down to a more ground state. And that can go to the electron transport chain, can go off to heat or non-photochemical quenching, or it can be given off as fluorescence. The first law of thermodynamics applies here. Matter can be neither created nor destroyed, and everything has to go somewhere. So if the chlorophyll molecule is excited, then that electron has to go somewhere. We would like it to go to photochemistry and the electron transport chain, but sometimes it doesn't. And we can measure the ratios of where it's going to help us understand what's happening in the leaf. We can think about fluorescence as a relationship of dissipation by all paths. Fluorescent yield is really by dissipations of all paths, which is another way to think about what I just said. It has to go somewhere. The energy has to be dissipated, has to be grounded. So it can go to heat, fluorescence, non-photochemical quenching, and there's also a small amount of basically housekeeping processes in the plant. So you can think about fluorescence yield either as everything has to go somewhere, or you can think about it in terms of rate constants, which is this particular slide. Some people prefer to think about it the first way, some people prefer to think about it the second way. Whichever one works for you to help you understand where those electrons are going, that's fine. So photochemistry and non-photochemical quenching are rate constants, which are adjusted by coefficients, and those coefficients are the proportion of open reaction center. An open reaction center is a reaction center in the plant that can take up an electron. So think of it as someone with their hands open and they're ready to catch a ball. That is an open reaction center. If the reaction center is closed, they're already holding the ball. And so they can't take another ball, it would just bounce off. And you can kind of think of fluorescence as the ball bouncing off of someone. The reaction center is already closed and the ball bounces off. And so we're able to see where that ball goes in terms of the chlorophyll fluorescence. So we're interested in measuring how many balls, how many electrons come back to us. If we put in light, how much comes back to us? How do we measure that? We measure it using something called pulse amplitude modulation fluorescence, or PAM fluorometry. The way PAM fluorometry works is we give the leaf an amount of light at a known wavelength and a known modulation rate. So modulation rate is how quickly that light is being turned on and off. And then we tell our electronics to pay attention only to this certain wavelength and this certain frequency. So I have a little video that one of my colleagues created. So we have a modulating beam. That's the thing that's flashing over here in the corner. We can do a saturating flash, which is where we give a very large pulse of light and we see what kind of fluorescence comes back. We can also turn on red and blue actinic light. Actinic light starts photosynthesis. We can do a saturating flash in a light-adapted plant. And we can do a far red pulse as well. And meanwhile, this entire time, this modulator and a detector. So this light is always flashing, it's always blinking. And the filter and the detector are paying attention to that flashing light. And they're not paying attention to any other light in the chamber. So if there's stray electrons and photons bouncing around, it doesn't matter. We are paying attention only to that light. We are focusing on that light. And then we can also use the combination of our modulating beam and our saturating beam to say, what is the fluorescence? So here's background fluorescence, how it's going in general, versus I gave it a very large amount of light, what came back to me. So here's some fluorescence parameters that you'll often see. As we start things going, then we just have F0 or F0, and this is just housekeeping fluorescence, just how things are going. We give a saturating flash in the dark, that gives us FM or maximum fluorescence. The difference between FO and FM is called FV. And then we go back to our FO. If we turn on the light, then we get FS, which is steady state fluorescence. And it is higher than FO because there's photochemistry happening and there are other photosynthetic processes going on, like non-photochemical quenching and perhaps a little bit of stress depending on the light level. If we then give a flash, we get FM prime. Prime means that we are working in the light. And the difference between the two is delta F. So delta F over FM prime is one of those things that we look at in terms of a light adapted plant. We can also give that dark pulse, that far red pulse. What that does is essentially forces all of the reaction centers in photosystem 2 to be open by making them all move to photosystem 1 using that far red light. And then that allows us to find out what the dark adapted light level would be in the light, but it's not quite the same. So it's called F0 prime. But that's the idea, is that we can get F0 without fully dark adapting our leaf. So here are some physiological parameters that we get from fluorescence and some things that you'll see in the literature. One thing I'll note is that sometimes literature will use different nomenclature. We use the nomenclature that is used in North America, not surprisingly, but occasionally you will, especially on older papers, you will see different nomenclature if the paper was published on a different continent. So one of the important things when you're thinking about looking at fluorescence is making sure you understand what exactly they're talking about. But so the efficiency of phi PS2, we can get two parameters here. We can get FV over FM, and this is a very common parameter. That's in the dark. And we can get phi PS2, which is in the light. We can also get non-photochemical quenching, or NPQ. We can get QL, which is the state of the reaction centers. And we can determine electron transport rate or electron transfer rate. You can also get electron transport rate from gas exchange. And so sometimes you'll see comparisons of the electron transport rate from fluorescence and gas exchange to see if they match up. Most of these parameters are ratios, as you can see, and they are dividing this by this minus that. And the other thing to keep in mind is that there are a few constants in here, which we will talk about a little bit later. We'll talk about how the constants can affect things. So one key assumption about fluorescence is that all of the reaction centers are open at the start in the dark, and in the light, all the reaction centers are filled. So if, for example, all the reaction centers are not open because your plant is not fully dark adapted, then your FV over